Hi everyone, and thank you for the introduction. And today I will be talking about navigating metabolic shifts in the transition period, immunity at the crossroads. So, as we are more aware about, the transition period is very challenging for all dairy cows because all of the changes that are happening at the same time. And it's because of this that even 25 years ago, this period was defined as the final frontier for dairy science and dairy production, with 30 to 50% of dairy cows having at least one disease during this period of time. And the question that we still have is why we are having this high incidence of disease and what is really triggering this. For sure, you might be aware that the immune system is playing a key role in coordinating this type of response. A concept that I would like to introduce you about is about how metabolic changes can affect immune response. So if we have different types of disturbance in cellular bioenergetics, this might be affecting the total function that that cell might have. And this is something that also happens with immune cells. So if we are having chronic and repeated activation of those cells, we can lead to an exhausted system. And overall, the function of those cells will not be as good as it should be. So what may happen is that if we have energy imbalance, this can impair health and increase the disease susceptibility that we may have, something that we also see in cows. And if we talk about immune cells, so which are the main changes in immune cell functions that are happening during the transition period? So we know that, for example, in the case of, I don't know if you can hear me, yes? Yeah. In the case of neutrophils, there is a decrease in their overall function. So we have a reduction in the oxidative burst, in phagocytosis, as well as in the formation of NEAT. So if we talk about monocytes, there is an increase in the pro-inflammatory response that these cells are going to have. And also, talking about macrophages, we have an increase in the infiltration of this type of cells into adipose tissue, and also an increase in the expression of pro-inflammatory cells. But now if we focus on what is happening on the adaptive side of the immune system, we know that in the case of B cells, there is a reduction in the production of IgG and also IgM. And if we talk about T cells, a very interesting research 20 years ago showed that there is a shift in the effector population that this type of cells have. And actually, we believe that this type of cells have a key role coordinating the whole immune response. So normally, if we are talking about the transition period, we refer as that as a cow in an immune suppressed state. But the question here is what is happening with this type of cells, the T cells? And mostly we are interested on T helper cells. So T helper cells, they are a type of lymphocytes that they have a key role coordinating the whole immune response. And this is because they are going to sense different signals that we have from the environment. And when they are in that resting state, they are going to be mostly relying on oxidative phosphorylation. And once they are activated, because they were present a specific antigen, they are going to be differentiated into a specific subset. So after that, we are going to have a metabolic switch in that specific cell, and the cell is going to start relying mostly on glycolysis, as well as oxidative phosphorylation. And this is because the cell should be able to support all of the proliferation and production of cytokines that we need to have the different function of the immune cell and activate the other cells on the immune system. So for you to have an idea, an activated immune system in a lactating cow consumes more than 1 kilogram of glucose in the first 12 hours of an acute infection. And also, as you may be seeing here in this picture, this metabolic function that the cell is going to have is going to be tightly related with the proliferation capacity and also with the cytokine production that these cells are going to have. And if for any type of reason these type of cells are not activated properly, this can contribute to have a wide spectrum of disease that has been widely studied mostly in humans. So again, the question that we have is which are the main changes that are happening on the metabolism of T helper cells and how this might be contributing to the high incidence of disease that we have during the peripartum period. So trying to answer that, this study, we have the main objective to evaluate the in vitro metabolic function as well as population dynamics of CD4 positive of T helper cells during the peripartum period, and also to evaluate the proliferation capacity of the cells. And our hypothesis were that those cells will exhibit a reduced capacity of their metabolic function, as well as a reduce in their proliferation capacity, and that we will see a shift towards a more pro-inflammatory state on these cells. So to evaluate this, we took blood samples from 18 cows at 20 days before calving, 3 days after calving, and 20 days after calving. I will call these periods dry period, postpartum, and early lactation. We isolate the PBMCs, the peripheral blood mononuclear cells, and we obtain an enriched population of CD4 positive T cells by doing magnetic sorting. And with that, basically, we did 2 different assays. So one was the cell proliferation assay with control and activated cells, and the other one was an assay to measure the metabolic function of those cells, also using cells that were controlled or at basal state and activated cells or stimulated cells. I hope not to scare you with this, but trying to explain a little bit more about how we measure the metabolic function of these cells. So to measure the cellular bioenergetics, basically we use a machine that is called Seahorse that allows us to have real-time measurements of the cell metabolism. So here we can see one of the outputs that we can have from the machine. This is the oxygen consumption rate. And basically here we can inject different types of compounds into the cells that are going to alter the metabolic function of the mitochondria. And with that, we are going to have different metabolic values. So here, at the beginning, we are going to get a first value of basal respiration, so it's the cell under no stimulation at all. After that, we are going to have a first injection that is oligomycin A that is going to block ATP synthase. And with that, we are going to have the value of ATP production. After that, we have the injection of BAM15, that is a mitochondrial membrane uncoupler. So now we are going to have a value of maximal respiration. And with that difference between maximal and basal respiration, we are going to have the spare respiratory capacity. There is a theoretical maximum that the cell have to respond under any type of stress. Finally, we have our last injection, that is rotenone and antimycin A, that these ones are complex 1 and complex 3 inhibitors of the inner mitochondria membrane. So now basically we are blocking all the capacity of the cell to produce ATP, and we are going to have the non-mitochondrial oxygen consumption. Moving into the results, here we can see the oxygen consumption rate that is going to be tightly related with the oxidative phosphorylation capacity of these cells. So here we have non-activated and activated cells. In red, we have dry period, in green, postpartum, and in purple, early lactation. So in the non-activated cells, there was a decrease in the oxidative phosphorylation capacity during postpartum, and it was higher in early lactation. But once we activate the cell, what is very interesting is that we found an increase in the oxidative phosphorylation capacity on those cells on both postpartum and early lactation, compared to what was happening in the dry period. So basically, oxidative phosphorylation capacity was increased after calving in the case of the activated cells. Now, moving into the other type of result, that is extracellular acidification rate, that is going to be reflected of the glycolytic capacity of these cells. So here in the non-activated cells, we found a differences between the dry period and the early lactation, and it was higher during early lactation. But once we activate the cells, we saw the same, that postpartum and early lactation were having an increase in the glycolytic capacity compared to what was happening during the dry period. So basically, the glycolytic capacity of the T helper cells was increased in both non-activated and activated cells after calving. As you may remember, we also have values of basal respiration. So here again, we have non-activated and activated cells. In this case, we found a tendency being higher in postpartum compared to dry period. But if we look at maximal respiration, we only found differences in the non-activated cells, being higher in early lactation and dry period. And if we do the difference between these two values, we can get what we call a spare respiratory capacity, that theoretical maximum that the cell have to respond. So here what is super interesting is that in the non-activated cells, we got a tendency between dry period and early lactation, and it was higher during early lactation. But in the activated cells, we didn't find any difference at all. And it's interesting because even though we have an increase in the glycolytic or oxidative phosphorylation capacity of the cells, we didn't have an overall increase of the capacity of those cells to respond in a case of an stress. Moving into the other assay, for the cell proliferation assay, overall, here we can see expansion index, division index, and percentage divide of these cells. And basically, what we found is that there was an increase in the proliferation capacity of those T helper cells during the postpartum period, that maybe this is corresponding also with increased metabolic function that they had. And using the supernatant from this cell culture, we measured the cytokine production of those cells. So here, what we measured was interleukin-6, interferon gamma, and interleukin-1 beta. So for the production of these cytokines, we found that in the case of interleukin-6, there was an increase during the postpartum. We didn't find any differences in interferon gamma. And in the case of interleukin-1 beta, we found a decrease during the dry period. So overall here, what we found was an increase in the production of pro-inflammatory cytokines after calving. And as take-home messages here, I would like to say that after calving, the T helper cells end up showing an altered metabolism and effector function, that this is also coincided with an increased metabolic stress that is trying to support the onset of lactation of these cows. Also, that maybe those metabolic changes and functional changes may be impairing the ability that those cells and that the immune system has to cope against any type of infection that may have during this period and mount an effective immune response. And also that if we try to understand better what is happening with those T helper cells that are key in coordinating our whole immune response, in this case for the cows, we might be able to enhance the immune resilience and reduce the disease susceptibility of dairy cows. And with that, I would like to say thank you for your attention, and I'm open to take any question that you may have.
Questions for Trinidad? Maybe a couple? Immunology, huh? Okay. Here we have one.
What's the difference between the 3 cytokines that you showed?
Okay, so between those 3 cytokines, we have interleukin-6. I'm going to let it like that. So we have interleukin-6. Interleukin-6 is a pro-inflammatory cytokine that mostly is going to be related with the production of acute-phase proteins. Interferon gamma is also, it can have both a pro-inflammatory and an anti-inflammatory role, but we didn't find any differences between those ones. And interleukin-1 beta is also a pro-inflammatory cytokine that has a key role like in calling all the macrophages to do the effector function overall. So what is important here is that all of them have a pro-inflammatory role, increasing the immune response.
Okay, any other questions? We have one here.
Thank you. I have a question on the, when you say like 30 to 50% of the dairy cows will have at least one disease after calving. So I was wondering, for all these parameters you measure, is there any difference in the healthy or healthier animals compared to the ones that with at least one disease, to see if there's any, if what you measure could actually indicate any health status?
Yeah. So in the statistical model that we use here, we incorporate morbidity as a variable here, and we didn't find any influence of that. But also I think that should be tightly related with how is that a specific disease that the cow may have. So probably if you can have like a group of cows that they have like a wrong transition period, or that they were really having any type of disease, and a group of cows that they were completely healthy during the transition period, you can see those differences. Because in this case, the cows that have like, for example, a retained placenta or those type of disease were not included. But I think that yes, that will be different as well.
All right, let's thank Trinidad.